All right, here we go. So here's where we're at today. We're going to be in week 3, protein synthesis video and guided notes. When you open that up, it should look like this. This is the link that's going to have the video on it. And we're going to go ahead and give that a watch. Right here.
The polypeptide. Every activity carried out by living cells uses one or more proteins. How are proteins made? The process called protein
All right, hold on. That's going crazy.
synthesis begins in the nucleus of a cell within a chromosome made of DNA, which stores the genes or recipe cells use to make proteins. The process begins when the section of DNA that contains the plans for a protein is copied or transcribed. The two strands of DNA separate, and free nucleotides pair with their complementary DNA bases. The new molecule formed is called RNA, which contains the base uracil, U, rather than the thymine, T, of DNA. The RNA is set free, and the DNA ladder zips up again. The RNA strand is called messenger RNA, or mRNA for short, because it carries the genetic message out from the nucleus to a cell structure called a ribosome, where the protein is built. The average cell of a mammal contains some 10 million ribosomes. The messenger RNA joins to a ribosome. The bases along the mRNA are grouped into sets of 3 called codons. Each codon matches a complementary set of bases called an anticodon. On another kind of RNA, transfer or tRNA. Each tRNA also carries one of 20 amino acids. Here, the tRNA carries the anticodon UAC with the amino acid methionine. The ribosomes assemble the amino acids together into a chain called a polypeptide in the order that forms a particular protein. As the first tRNA floats away from the ribosome, it leaves behind its amino acid. The ribosome moves along the messenger RNA. Each transfer RNA brings along its specific amino acid. The amino acids are assembled one at a time in the right order. Protein synthesis ends when a special stop codon is reached. The string of amino acids has become a polypeptide chain. The ribosome then releases the mRNA and the polypeptide. The polypeptide chain folds into a 3-dimensional shape that is unique to each protein's structure and function.
Okay, so that's the first time through. So hopefully you guys caught a few of those. All right, that go with it. So every activity carried out by any living cell uses one or more proteins. All right, so then we can I'll go through. All right, so when I play it through this time, I'll stop it each time it gives you an answer. All right.
Every activity activity carried out by living cells uses.
I don't know why it takes a minute to buffer. All right, so it uses one or more proteins. There you go. That's one of them.
One or more proteins. How are proteins made? The process called protein synthesis begins in the nucleus of a cell within a chromosome made of DNA, which stores the genes or
All right, the genes are the recipes of the cell.
recipe cells use to make proteins. The process begins when the section of DNA that contains the plans for a protein is copied or transcribed.
Okay, there's another one. Copied or transcribed.
The two strands of DNA separate, and free nucleotides pair with their complementary DNA bases. The new molecule formed is called RNA, which contains the base uracil, U, rather than the thymine, T, of DNA.
Okay, so there's another one called RNA. This is a single-strand molecule that contains the base uracil rather than thymine. We talked about that yesterday, how RNA is a different base pair, and that is U instead of T.
The RNA is set free, and the DNA ladder zips up again. The RNA strand is called messenger RNA, or mRNA for short.
Okay, mRNA, we talked about that yesterday, how this one is the one that's allowed to leave the nucleus in order to go out into the cytoplasm and be transcribed.
Because it carries the genetic message out from the nucleus to a cell structure called a ribosome, where the protein is built.
Okay, so like I said, it carries it out to the ribosome, which is one, and the protein is built.
The average cell of a mammal contains some 10 million ribosomes.
Okay, average cells, 10 million ribosomes.
The messenger RNA joins to a ribosome. The bases along the mRNA are grouped into sets of 3, called codons. Each codon matches a complementary set of bases, called an anticodon.
Okay, so set of 3 right here, like this one is UAC, is the anticodon, which is matching the codon, which is the first one, UAG. So these codons have complementary pairs called an anticodon. This protein right here brings over this amino acid MET, which is used to build.
On another kind of RNA, transfer, or tRNA. Each tRNA also carries one of 20 amino acids.
There are 20 different amino acids.
Here, the tRNA carries the anticodon UAC with the amino acid methionine. The ribosomes assemble the amino acids together into a chain called a polypeptide in the order that forms a particular protein.
Okay, so ribosomes is some like this MET-SER. Those are the amino acids. Now, if we had time in a full biology class, we would probably memorize and kind of know those pretty well. and talk about what proteins, what polypeptide chains make which protein, and so on and so forth. So I know this is kind of confusing, but when your body needs something, it will send out a messenger to tell the nucleus in order to create these polypeptide chains, which makes proteins, which it will serve functions that you need. Like if you need to stop bleeding somewhere, there's a protein that helps with that. So then your body would start making these proteins in order to get your body to stop bleeding.
As the first tRNA floats away from the ribosome, it leaves behind its amino acid. The ribosome moves along the messenger RNA. Each transfer RNA brings along its specific amino acid. The amino acids are assembled one at a time in the right order. Protein synthesis ends when a special stop codon is reached. The string of amino acids has become a polypeptide chain. The ribosome then releases the mRNA and the polypeptide. The polypeptide chain folds into a three-dimensional shape that is unique to each protein's structure and function.
Okay, so the stop codon is a codon that pairs for something that says stop, like, hey, stop transcribing right here. All right, so when it stops, then the protein folds into a three-dimensional shape, which is based on whatever the structure and function of the protein is. So different proteins have different shapes to allow for different uses in the body. So that should have helped with these notes. All right. Any questions so far? Okay. All right, so let's take a look at this exit ticket. So this exit ticket is pretty pretty good. It looks complicated, but once you get started on it, and you kind of learn how to use this here, this table, it flows pretty quick. All right, so this will be a question that you'll see tomorrow on your end of the week assessment. What it does, it takes different codons and it shows you what amino acid that it codes for. So I'm going to transfer this over to my other whiteboard, which helps you guys help me draw on things so I can show you how to use this. All right. So we're going to be using this table in order to answer this first question. So mRNA codons are here, and we're going to use it to find which amino acid it is. All right, so let me transfer that over. Okay, so you guys should be able to see this codon chart side by side with the amino acid chart. All right, looks like it's working. All right, so that's good. All right, so here's how this works. You're going to have, this is going to be your first step here in green. Okay, so your first step is going to be here using this column. Then you're going to use the second letter in the string to find the second box it's in here. And then you're going to use the third row to land on the specific amino acid. So, for example, this one, all right, I'm going to change colors around so they stick out. All right, you have the mRNA codon G. So I'm going to start G right here. So that means I'm going to be in this row only. Highlight that for you. Okay, next, U. So then I'm going to look at the column up here, U. So then that brings me into this box here. So that means this box is where I'm at next. That means this is the only box I'm looking in. All right. Next, and finally, we're going to tackle A. So A is the last one. So I'm going to use this box here to find A. So if I follow all the way over, it's here. So this is the codon I'm looking for. All right. And the amino acid is what's in lowercase. So it's that VAL is what I'm looking for. So that means the amino acid here is VAL. So those are the steps that we take in order to find the amino acid. So let's do another one. Okay. Going to, which one right there? All right, so first up, we got U. So I'm going to start here. That means I'm only going to be in this row of U. Next, I have another U. So that means my next one is going to be in here. So that means I'm only going to be looking in this box here. Finally, I have a third U. And then that means I'm going to be using this one here. So if I follow that over, the codon UUU codes for the amino acid PHE. So that's how you find the amino acid. All right. Let's do another one. CAC. All right, so we got C is up first. So I'm going to be looking in this row. A is next. So I'm going to be looking in this column. And then finally, I'm going to be looking in the row of C. So I'm here, here. So that means I'm in this box here. So the last one is C. So if I go over, it's right here. So you'll see that this in here, CAC, matches the codon, CAC. So then my amino acid would be HIS. A lot harder to do with a finger. Okay, so anybody lost? You guys want to try number 4? So try number 4 and type the amino acid in the chat. So if it was HIS, you would just type in HIS in the chat. So we're going to try UAA in the chat. Excellent. All right. So you should have got a stop. All right. So you got U, and then AA is right here. So UAA is a stop. So this is a stop codon. This is what they talked about in the video. Let me write that in. So that what happens is when these are built, that UAA is one that they use to stop the process. So otherwise, this process would go on indefinitely forever. So the stop codon tells the ribosome to stop making protein. And it disconnects there. Kind of like, all right, we're done here. We got what we need. We're stopping. So if you look around, there are 1, 2, 3 stop codons. Okay. Oops, wrong color. Here's a stop, there's a stop, and this is a stop. So out of all these combinations, only 3 of them tell the protein to stop. Okay, so it looks like you guys get this. That's awesome. Okay, so let's see here. See how much time we got left. All right. So I'm gonna give you guys, I don't know, maybe like 5 more minutes before I show you how to do this one. So I'll let you guys work through numbers 5 through 10, and then I'll help you out with that last one. And give us about 5 minutes to do that before I jump back in.
want you to ever speak to me again. No way. I'm torn.
Hey, Nakaya, we're on Schoology in the protein synthesis video and guided notes part. We are going through the exit ticket right now. So if you want to get it pulled up, I'm going to finish going through here in a minute. All right, guys, I'm going to knock out this last part with you guys, show you how to do it. I'm going to do that first section. All right, so this bit down here is kind of a wrap up or a combination of everything we've gone over. Let me get it right. All right, so what this is an example of is the DNA sequence from the DNA strand inside the nucleus. It would code to an mRNA strand, which would leave the nucleus to make an amino acid. So this is kind of like the process of it all, how it works step by step. All right, so here we go. All right, so the DNA sequence. All right, you got to remember what each part in the DNA codes to. All right, so G codes to C. So you keep forgetting I'm not on the drawing part. All right, so G codes to C, and vice versa. So that means C would code to G. Let's see if I can do this little arrow. And for DNA, A codes to T, and T codes to A. But in mRNA, there is no T. That means if you see a T, all right, you're not going to A. So let me revise this. So T can code to A, but A will code to U instead. So if you're copying this to another DNA strand, it'd be different. But the complementary mRNA is not going to have any T's in it. So this right here, row, should not have any T's in it. So G will go to C. C will go to G. And T goes to A. So then you take this right here and you take it up to your table up there. So you need to start with C. So I'm starting C there. Go to G all the way over here. And then I'm going to be in the A row. So this right here is the protein I'm looking for. So the amino acid, amino acid, not protein. So the amino acid is ARG. So then you're going to continue that idea through these two boxes as well. So you're going to look up ATA. A codes to U. It does not code to T. If you look up here, you will see no T's in this box up here, your codon box. You're not going to see any T's. That's how you know if you put a T down, you messed up. So remember, A goes to U, T goes to A goes to U. So this one should be U, A, U. And then you go up here and find that. U, A, U would be TYR. And then you would finish out and do this last one. So it's kind of the three-step process. It's like the DNA leaving, making an mRNA strand, which leaves the nucleus and makes an amino acid. That's how this one works down here. So it's like a three-stepper. All right, any questions on that? All right, so that's all I got for you today. If you need any help, stick around and ask me, and I'll be here. Remember to submit this on Schoology. Submit when you finish and you fill it all in, submit it on Schoology.